Thank you so much. Appreciate the introduction, and I'm really happy to be here to present some of the work that we've been doing. As we heard in the introduction, I'm at UC San Diego in La Jolla, California, United States. I'm jointly appointed in the departments of both pediatrics and Scripps Institution of Oceanography, where I'm the deputy director. That relationship between clinical science and environmental science pervades nearly everything I do in my lab. And we have an exciting, diverse array of research programs that span both environmental health and human health. I'm going to be talking predominantly today about human health in the context of both the built environment and precision medicine. So I just wanted to start out, the Society for Applied Microbiology was rebranded Applied Microbiology International in 2022. And I'm now the president of that society. I want to highlight the value of being a member of a society and publishing in society journals, such as the journals for the American Society for Microbiology or Applied Microbiology International. All the money that you provide to the societies comes back to you through grants and funding, educational and professional development opportunities, networking and events, etc. So I strongly recommend you join societies like Applied Microbiology International. And if you want to find out more, you can let's go to this link, appliedmicrobiology.org. It's an international organization with members in over 75 countries at the moment. Okay, so I want to start by talking a little bit about the microbiology of the built environment. These are the microorganisms that inhabit the spaces in which we live and work. We each emit around 38 million bacterial cells into our immediate environment every hour, and some 7 million fungal units, and probably 10 times as many viral particles. This environmental release that we provide to the environment also comes back to us when the bacteria that have survived their trip outside our body into this sparse, dry, hostile environment to them come back to us. This is often considered to be an epidemiological investigation. So, for example, pathogens leave our body, land on the surface, and they're transmitted to somebody else or through the air. But there are many, many other microorganisms that are not pathogenic that we are just starting to understand how to interact with and explore. What we do in our research is try and understand these dynamics and drive the research forward to create opportunities to intervene. There are many factors that are associated with airborne fomic pathogen transmission. So we know, for example, that microbes from the air are inhaled into our lungs, and this can include things like rhinoviruses, SARS-CoV-2, which everyone knows about, Bacillus anthracis, Legionella pneumophila, and fungi. But we also emit those organisms back into the environment. And the other route is through touch. So skin-associated or mucous membrane-associated interaction can acquire organisms such as C. diff, and that should say Clostridioides difficile. I apologize for the old genus name. Staphylococcus aureus, Pseudomonas putida, and fungi and viruses, and we transmit them to the environment through our skin. What we wanted to understand was how to explore that environment to improve health of the occupants. And this comes back all the way to Florence Nightingale, who was a nurse during the Crimean War in the 1800s. And she explored ways of improving the built air in hospitals by opening windows. So she is quoted as saying, to keep the air he, the soldier, breathes as pure as the external air would significantly improve the health of those occupants, of those patients, and reduce the mortality on her wards. So while opening windows to allow fresh air is valuable and can reduce the abundance of airborne pathogens, it's not always possible. And so we have been exploring recently strategies to both control the pathogenic microbiome of buildings and also to promote the establishment of beneficial microorganisms in these settings. One approach that we've explored is applying bacteria such as Bacillus subtilis to hospital services and examining the opportunity of this living organism to outcompete or metabolically deactivate certain pathogens. So, for example, here, when we take a cleaner that doesn't have any bacillus in it, or a control cleaning product, comparing it to a cleaner that has Bacillus subtilis spores embedded in the liquid, we can see that there's a significant reduction in richness and a significant decrease in the abundance of many different types of pathogens, such as Staphylococcus, Streptococcus, Mycobacterium, and E. coli Clostridia. We've also been exploring how to add bacteria into the surface materials themselves. We are printing the bacillus spores and many other types of bacteria directly into surface materials that then contain and hold those bacteria. These images do not show bacteria associated with the particles, but show what these nanomaterials are structured like to contain cells. So the fabrication of these 3D constructs is done by extrusion-based printing, which it can be done at a room temperature and can be done to improve the bacterial viability. And these scaffolds are spatially controlled microarchitectures that allow for bacterial growth and sustainability by providing both the nutrients and pockets of moisture associated with materials. And once they're seeded with a type of microorganism, they are often impervious to incubation or inoculation or by other types of organisms. So these structural materials we are exploring as a way of directly introducing microorganisms into these environments. And you can see a mock-up here of a particular type of surface material. This is a living surface material printed with bacteria in it. Here's the actual reality. The white area is all ceramic that has microorganisms in them, and behind that mesh, we have behind this living ceramic on the outside, we have a scaffold which allows for airflow to pass through the material, providing an innovation in HVAC systems. And if you're interested, Professor Richard Beckett, who's one of my colleagues and collaborators at University College London, and he's an architect, has written this book on probiotic buildings or probiotic architecture, probiotic cities. I strongly recommend reading this by Richard Beckett. So understanding how to manipulate and improve the health of the building outside is one thing. We want to understand also how to improve the health of the body inside. And so I always come back to, what are we trying to do? Well, when we think about the microbiome in our bodies and on our bodies, we have to understand how it gets there, how it's controlled by the body, and what impact it can have on health. So microorganisms interact with our immune system in a very, very specific way. The immune system evolved to manage the microbial garden living inside and outside our bodies. And so the immune system actually manipulates the growth of particular microorganisms to promote the growth of bacteria, which provide our bodies with beneficial services. So in many ways, the immune system is more like a national park service or maybe a gardener. It's there to manage that microbial garden to improve our health. When something goes wrong with the microbiome, it can prevent the release of the products, those beneficial products, which can actually damage the immune system. And when the immune system's damaged, it can also lead to a lack of control of the microbiome, causing a runaway growth of pathogens. And we can see that kind of scenario when we use this infographic from Mariana Bindlos's team. It's an old graphic, but it's really instructive. So here we have a cutaway of the gastrointestinal tract. On the left, we have a system that's in balance, and on the right, we have one that's out of balance. What do we mean by homeostasis or balance? Well, the blue in the gastrointestinal lumen reflects a low oxygen potential, so not much oxygen around, or zero oxygen in some cases, and that allows the growth of obligate anaerobic bacteria. Many of these obligate anaerobic bacteria ferment fiber, and in doing so, they produce short-chain fatty acids. These short-chain fatty acids are actually fuel substrates for the colonocytes that line your intestine. The colonocytes that line your intestine are replaced from the bottom of these ducts about every 7 days. So your entire gastrointestinal lining is being replaced about every week. And these short-chain fatty acids fuel the growth and maturity of these rapidly changing cells. So through a beta oxidation pathway, which consumes oxygen out of the bloodstream, we see an improvement in the integrity and the tight junctions of this gastrointestinal membrane. These short-chain fatty acids also are fuel for the immune system, fueling T regulatory cells, which can help to drive down inflammation and maintain immune homeostasis inside the body. So this is a self-balancing mechanism. This healthy, mature lining consumes oxygen, which stops the oxygen from getting into the gut cavity, which allows these obligate anaerobic bacteria to grow, so they can produce short-chain fatty acids to allow the cells to consume oxygen. Right? It's a partnership. It's a cycle. When you disrupt that cycle, let's say we pump the gut full of a non-absorbable antibiotic like vancomycin, it kills off a lot of these obligate anaerobic bacteria. But it can allow the growth of facultative anaerobic bacteria who have antibiotic resistance. Many of these facultative anaerobic bacteria produce things like flagellin, to allow their flagellas, or lipopolysaccharide, or even hydrogen sulfide, which can, all these products can be pro-inflammatory. These bugs also do not ferment fiber, or very rarely do they ferment fiber, and they do not produce short-chain fatty acids. So now this rapidly growing cellular membrane doesn't have enough fuel to allow it to mature, and so you've now got an immature gut lining, which can have holes in it and allow particles to get inside the body. They also don't consume oxygen because they then drive their processes through anaerobic glycolysis, and that oxygen now can get into the gut cavity, raising the oxygen potential. So not only do we have bacteria which produce products which cause inflammation, we also have no products being generated that can help the gut lining to become mature or fuel our regulatory T cell anti-inflammatory response. So now you have a negative feedback cycle which prevents the regeneration of homeostasis. The best way we've found in our animals to go from here, dysbiosis, over to homeostasis is through diet, is to allow the consumption of fiber, polyphenols, indoles, which can help to support the gastrointestinal microbiome and allow the good bacteria to grow back. But dysbiosis, this imbalance in the microbiome, once you understand it as a mechanism, you can really start to figure out ways in which it affects our bodies. So in this one study, we looked over time, taking samples of mice every 3 hours from their entire gastrointestinal tract for 48 hours, and understanding how that dynamic over a 2-day cycle changed their microbiome and changed their hormonal levels. So in this one study, when we took germ-free animals, it really didn't matter if we gave the animals a low-fat, high-fiber diet or a high-saturated fat, high-sugar diet. As long as we provided the same amount of calories, in the absence of bacteria, the animal will stay lean. Obviously, if you provide too many calories, the animal will gain weight. But in the basic, it really doesn't matter what we're feeding the animal. The metabolism, the metabolic health of the animal will be approximately the same. Oh, sorry. If we have animals with bacteria in them and we feed them a healthy, high fiber, high polyphenol diet, we'll see normal microbial oscillations, so changes throughout the day that are repeatable day to day in the microbial community metabolism. And these normal microbial signals allow for normal circadian function in the body, and the animal maintains a lean phenotype. But if we give them a high saturated fat, high sugar diet, we start to see aberrant microbial signatures, an increase in those bacteria, the facultative anaerobic organisms, especially during the early morning. They can produce high levels of hydrogen sulfide, which gets into the bloodstream and can actually disrupt clock gene transcription in the liver. This now decouples the brain from the liver in terms of the perception of time, and that decoupling leads to hormonal activation by the liver, which can lead to a significant increase in food energy storage and therefore adipose tissue generation and an obese phenotype. So, this high saturated fat, high sugar diet tends to increase those facultative anaerobes, increase hydrogen sulfide production, altering hepatic hormone activity and increasing adipose tissue production and appetite in the animal. We wanted to explore if this same function exhibits in humans. And so for the last 10 years, we've been following a cohort of people of African descent, over 2,500 people from 5 different countries: Ghana, Jamaica, the Seychelles, South Africa, the United States. And this is a project led in my lab by Gertrude Eckler-Mansar, who's one of our very talented project scientists. So in this study, we can see, if you look at just alpha diversity on the x-axis, Ghana has the highest alpha diversity, followed by South Africa. Both the populations we examined there are living in the rural communities. Compared to Jamaica and the Seychelles, the United States tend to have lower alpha diversity and live in very much more urbanized and highly processed food consuming communities. And when we associate these alpha diversity metrics with health outcomes, we can see, for example, that irrespective of which country you live in, higher alpha diversity is associated with low blood glucose levels, low blood pressure, normal BMI, and a lack of hypertension. So we can see that there is an association between microbial diversity and the metabolic health. What's interesting, though, is if we look at the particular types of species of organisms that are associated with that, irrespective of country, and again, you can predict country extremely well, AUC of 0.97 in the ability to predict which country a person comes from. We aren't able to really accurately predict obesity. AUC is about 0.65, 66, or hypertension, but we can predict blood glucose levels very well. We can predict diabetes very well, and obviously birth sex is pretty good. So 0.92 for diabetes is a very, very successful average. We also know that dysbiosis can affect how your body responds to other things. So, for example, here I'm doing a gastrointestinal resection. So I've removed a bit of the gut. That allows oxygen into the gastrointestinal cavity, which kills off a lot of bugs. And then we pump the body full of antibiotics during surgery, which kills off a lot more of the bugs. Now you have a Wild West environment, a Mad Max zone, where only a few very resistant organisms are surviving. And when we stitch the gut back up, the body starts to suck nutrients out of the gastrointestinal tract in order to repair itself. And this starves the bacteria, causing them to release a new phenotype, which allows biofilm formation and the production of collagenase, an enzyme which breaks down the collagen to allow the bacteria to get nutrients. And this can cause an anastomotic leak, where the gastrointestinal contents leak out into the gut cavity. We used to assume that the bacteria that caused this were bacteria introduced during surgery. However, it turns out that that's not necessarily the case. Nearly every single patient we looked at had the organisms in their gut when they came into hospital. And we think it's associated with diet. So, for example, what you eat, where you live, and your exposure to furry pets, for example, can shape your microbiome. But what you eat tends to increase the abundance of collagen-lytic organisms in your gut. These collagen-lytic organisms are some of the ones that survive gastrointestinal lavage through a polyethylene glycol consumption and oral antibiotics, survive IV antibiotics, and also survive surgery, and then start growing on the gastrointestinal lining. Now, if everything works, those bugs should go back to normal, to the way they were before after surgery. But invariably, they don't, and they can cause surgical complications. We demonstrated using an animal model that a Western diet plus antibiotics can significantly impact the likelihood of these collagen-lytic organisms altering colorectal cancer tumor development inside the gut. So in here, we again give the animals a Western diet, and then antibiotics, oral, ad libitum through liquid, and then we do a colorectal resection. What we're expecting is these collagen-lytic bacteria to grow around that anastomosis and allow colorectal cancer cells, which are present inside the animal after we injected them into there, to start to grow on the membrane and cause a perianastomotic tumor around that site. And what we see is that collagen-lytic E. facialis, this is Enterococcus facialis, which can produce collagenase in the distal colon, is significantly elevated when the animal consumes a Western diet. So the very consumption of a saturated fat, high saturated fat, high sugar diet increases the proportion of E. facialis in the animal's gastrointestinal tract. What's interesting, though, is the ability of the mice to form tumor is also associated. So if they eat a high fiber chow and they can just have antibiotics and surgery, the likelihood of a perianastomotic tumor forming is only 10%. If they consume a high fiber chow with antibiotics and surgery, and we introduce this collagen-lytic E. facialis in there, it goes up to 30%. If we give them a Western diet, antibiotics and surgery, in the absence of the collagen-lytic E. facialis, the abundance is about 60%. If we give them a Western diet, antibiotics and surgery, and this collagen-lytic organism, nearly 90% of all our animals generate perianastomotic tumors. So a Western diet promotes the growth of bacteria, which not only can damage the gut during surgery, but also can increase the risk of colorectal cancer. We also know that the microbiome can play a very significant role in mental health. So neurological disorders are often associated with diarrhea and constipation. We've known that for years. And changes in the microbiome are often associated with neurodevelopment, with neurodegenerative disorders such as Alzheimer's and Parkinson's, and also mood and behavior, which can vary throughout our lifespan. We also know that antibiotics are often linked to behavioral phenotypes like depression and anxiety, but interestingly, antivirals and antifungals are not, suggesting that it's the killing of bacteria in the gut which has the biggest association with the onset of symptoms. We also know that we can take clinically depressed people's fecal microbiome and do an FMT into a mouse model, and those animals will show depressive-like behaviors. And I'll talk about what those are in a minute. But non-depressed people's microbiome doesn't demonstrate the same phenotype change. So there's something, again, about the bacteria that are present in a clinically depressed person's gastrointestinal tract that appears to alter phenotype. We know that there's 3 different potential mechanisms: modulation of neurotransmitters. Bacteria in the gut produce a lot of the precursors of serotonin that's then produced by cells in the intestine. We know they also produce GABA and norepinephrine and dopamine. We also know that bacteria can alter inflammation, which is a major associated signature along with depression. So inflammation triggers cortisol stress responses through the HPA axis, and cortisol levels in the blood can cause levels of anxiety and stress. We also know that direct signaling can happen. So receptors on the vagus nerve or efferent spinal pathways have been shown to be triggered by bacterial metabolites. All of these pathways are also existing drug targets for most central nervous system diseases, but we think the microbiome may play a significant role. So we did one study where we took fecal microbiome transplants from Sprague Dolly rats, which generally tend to not show a depression-like phenotype, into fawn-hooded rats, which tend to show a depression-like phenotype. So, for example, here are 2 tests that we use. Pardon me. 2 tests that we use to associate or identify depression. First one's the forced swim test, whereby we place the animal in a jug of water, and the animal that really tries to get out and spends all of its effort trying to get out is considered to be less stressed than an animal that just gives up and just lies there floating, giving up on life, right? So in here, the floating time in terms of seconds is significantly higher in the fawn-hooded rats compared to Sprague Dolly rats, as we would expect. The Sprague Dolly rats spend less time floating and more time trying to get out. Interestingly, when we take the microbiome from a Sprague Dolly rat and we put it into a fawn-hooded rat over here, we see a recapitulation of that phenotype. So now the fawn-hooded rats fight for life. The other test is sucrose preference. This is what's known as a hedonistic test. So the animals will show a preference for sugar. Those that show a bigger preference for sugar tend to be more hedonistic and less depressed. And we see this in people as well. People tend to stop getting pleasure out of things which gave them pleasure before, like sugar. So in here, a fawn-hooded rat has a much lower sugar preference than a Sprague Dolly rat. But when we take that fawn-hooded rat and give it a Sprague Dolly microbiome, its sugar preference goes up dramatically. So these 2 phenotypic tests demonstrate an improvement in the symptoms of the animal with a fecal microbiome transplant between rats, which is highly effective. We also show that those fawn-hooded rats that get a Sprague-Dawley microbiome have significantly elevated levels of serotonin in the hippocampus, significantly elevated levels of norepinephrine, again, similar to the Sprague-Dawley rat originally, and also lower interleukin-1 beta and interleukin-17A, which are markers of neuroinflammation. So they got higher levels of beneficial serotonin, neurotransmitters, and lower levels of inflammation in the brain. So what do we think is happening here? Well, one thing we did find when we looked at the metabolomics of the bloods of these animals was that fawn-hooded rats generally have a gut microbiome which produces an elevated level of carbon-rich metabolites. These carbon-rich metabolites tend to end up in the brain and tend to trigger inflammatory responses. When we swapped out the microbiome in this fawn-hooded rat for what a Sprague-Dawley microbiome, we see a reduction in species like Dialista, and this microbiome, the new microbiome, tends to produce nitrogen-rich metabolites. So we get a reduction in the carbon-rich metabolites in the bloodstream, an increase in nitrogen-rich metabolites in the bloodstream. And these nitrogen-rich metabolites, which are elevated, like serine, in the hippocampal region, also seem to appear to drive down osmolytes, drive down inflammation, and increase serotonin, norepinephrine, and dopamine production in the brain. So there is a direct association here between the microbiome and physiological responses. We can also see this in people. So in this study from 2018 that we did, we looked at the functional magnetic resonance imaging in patients with major depressive disorder, and we see a direct correlation between the GABA-mediated response of connectivity between the prefrontal cortex and the abundance of Bacteroides in the gut. So certain types of Bacteroides species appear to be elevated in people who have no neurological symptoms of depression, and depleted in people who have elevated neurological symptoms of depression. We know that Bacteroides do synthesize gamma-aminobutyric acid in the gut, and we know that they do have an anti-inflammatory phenotype. So we took some of these animals and working with very talented postdoc in my lab, Romy Das, and a very talented new graduate student in my lab, Marisol Dotti-Dothard, who's a phenomenal scientist. We wanted to explore whether these bacteria could have a big impact upon depression in animals. So we have an animal model of depression called social defeat. And these animals show when they are socially defeated by a dominant male for a long period of time, tend to show a significant reduction in that sucrose preference. So it's an animal that's sort of significantly stressed. We can rescue that phenotype when we give the animal ketamine, which is a very now common antidepressant. But we can also do it when we take this bacteria, this Bacteroides strain, which the company HoloBiome has started to try and commercialize and identify, and add this one bacteria, and it has a similar rescuing effect to ketamine, which is potentially interesting. I am on the scientific advisory board of HoloBiome and do have a financial interest in the company. But this result's fascinating. We also know that the microbiome can have a very significant impact upon the immune system and health and disease, even in the lung. So in this one study, we looked at chronic obstructive pulmonary disorder in a discovery and validation data set from 2 cities in China by looking at the metagenome of sputum and the metabolome of the sputum and the host RNA-seq of cells associated with that. And we also did the proteome. In this study, we found a number of different things, but the super cool stuff was that neutrophil predominant chronic obstructive pulmonary disorder had a reduced abundance in certain species of lactobacillus in the lung, which resulted in a reduction in the level of indole-3 acetic acid synthesis by those bacteria in the lung. And this was in turn linked to perturbed IL-22 signaling and epithelial cell apoptosis. So in the COPD airway, they have less lactobacillus, therefore less indole-3 acetic acid, then a deactivation of the AHR1 mediated response, which would lead to an increase in IL-22. Instead, there's a decrease in IL-22, which leads to a significant reduction in the integrity of the epithelial membrane in the lung, which causes emphysema, inflammation, and apoptosis, and a reduction in lung function. We can see that we're able to rescue this by adding in a mouse model of lung damage species of Lactobacillus salivarius or Lactobacillus aureus into the lung. And both organisms, either in isolation or together, have a significant improvement in the histopathological score of the lung tissue of these animals. So the lung tissue becomes less damaged. Also less inflamed, less IL-6 and less IL-17A, so less circulating pro-inflammatory cytokines. We also have been doing this in a fun study we did with dolphins just out here in the bay. There's a population of semi-wild but trained dolphins that work for the National Marine Mammal Foundation through the US Navy program. And we wanted to see if the microbiome of the dolphins could improve gastrointestinal symptoms of what essentially is IBS, so, irritable bowel syndrome in the animals. We don't cause it, but they do get it sometimes, and it's very uncomfortable, causes diarrhea. So we used a fecal microbiome transplant method to try and examine this. And we did demonstrate in many animals that they saw clinical improvements in their dysbiosis-related symptoms following FMT treatment. And also the microbiome diversity significantly increased in the recipient post-transplant to resemble that of the donor. So we do see shift in taxonomic composition in these animals. So we can even do fecal microbiome transplant in dolphins to treat their diseases. We also know that microbes can be involved in microbial decomposition. In this one study we did with Rob Knight's lab, we were able to identify microbes that were associated with decaying human corpses and the level of microbial diversity, demonstrating that the microbial community structure can actually accurately predict the post-mortem interval. I put this study in here not to be too weird, but to demonstrate that there are many, many different ways in which we can use the microbiome to explore human life and death. So what have we been doing? Well, we've been exploring the microbiome in some interesting ways for many, many years. On the left, we have a pediatric Bristol stool chart. So type 1 of stool, it looks like rabbit droppings, separate hard lumps like nuts. They're hard to pass. It's close to constipation, right? And then you have all the way down the bottom, it's like gravery, watery, entirely liquid. Essentially, that's diarrhea, and you have everywhere in between. So we've been looking at stool consistency for many, many years, hundreds of years, in fact, to try and diagnose health. All we're really doing now is taking this same kind of information and weaponizing it with microbial DNA signatures to truly understand that dynamic. So we call this analysis Microbiome-Wide Association Studies. These are akin to genome-wide association studies, where we look at an allelic genetic variant in a population and associate it with health and disease. Instead, we're looking at the microbial genomes, the microbial metabolites in that community, and see if they are associated with disease. We found some really interesting things that way. So, for example, we know that microbes produce things like acetate, which is associated with satiety, or indoles, which can be associated with improved lung function or reduction in asthma. We also know that the bacteria in the gut can alter acetaminophen to make acetaminophen sulfate, which can actually damage the liver. So this is the reason acetaminophen is probably one of the most dangerous drugs on the market in the US, despite it's completely freely available all the time. We also know that the microbiome can consume chemicals that our own body produces, like bile acids, to make secondary bile acids, and that the balance of these bile acids is really important for the health of the gut. So doing these microbiome-wide association studies, we can find many different mechanistic analyses in the system. And again, the American Gut Project, which is relaunching soon, allows us to gather this data using people donating money to get their microbiome sequenced, which can create, and I think we have over 30,000 people with their microbiome sequenced. So it's a freely available public data set which you can use for discovery. But one thing we have found is that one sample from people, it really isn't enough to understand their variability due to the inherent dynamics of the microbiome during the day, and also day to day or week to week, or with changes in your life's activity. We see that time is really important. And this one very, very simple graph from one of my ex-graduate students, a very talented associate professor, Sean Gibbons, demonstrates that the number of time points you sample in an individual significantly improves the ability to detect a biomarker, or should I say, reduces the variation in our ability to detect that biomarker. So the more samples you have in a person, the better your ability to actually detect something which will have an impact upon that person. So why is time so important? Well, unlike things like height, weight, and age, which are pretty low dimensional, they're single numbers, right? And they're pretty static. They don't really change much week to week, on the whole. Or blood pressure and insulin, which again are low dimensional, but they're highly dynamic. They can change hour to hour in terms of their concentration in the blood. Or genetics, which is pretty static overall, but very highly dimensional. There's 28,000 genes in the human genome, so it's got a lot of information in it. The microbiome is both high dimensional and extremely dynamic. So it causes, it's much harder to use it to identify effective biomarkers. So to discover precision microbiome processes, we need a big cohort size and a lot of frequent sampling. And so we are launching, as was mentioned in the bio at the beginning, a new program, the Nutrition Precision Health Program for the NIH. It's a $170 million program to examine how people respond to food. And in this study, we're actually leveraging that time element to really understand how time associates with our ability to digest or and analyze food. So we're actually collecting samples every single day from these people. So we have 3 modules in this investigation. Module 1 is a characterization of 10,000 people, looking at their food recall, body performance measures, physiological microbiome measures, blood chemistry, etc., on their normal diets. And then in module 2, we're taking 2,500 of that group, so a quarter of them, and putting them on 3 crossover prescribed diets at home. So they come in, they're the regular diet, they go for 14 days on diet 1, wash out, 14 days on diet 2, wash out, 14 days on diet 3. And all the while we collect tons of data on them. And then for module 3, which we just started, we are doing a longitudinal analysis of 500 people on the same 3 crossover diets, but in domicile clinical centers. So we have complete control over everything they do, right? So this, we believe, has statistical power to assess the impact of a med-dash diet. This is an example of our daily menu for a med-dash diet, which is a heart-healthy diet based on a Mediterranean diet. We also have a high-fat, low-carbohydrate diet, which is akin to a ketogenic diet, but we want to understand how people respond to this. And then we have the SAD diet, or the standard American diet, which is high saturated fat, high sugar. And so we put people on these 3 diets, and so each person is their own control, having a crossover design that's randomized to allow us to detect how diet influences both human health through changes in the microbiome. So there's a huge amount of data going into this study, including wearables, dietary assessments, fecal metagenomics, and metatranscriptomics, using a really new technique that provides quantitative data that was pioneered by Carsten Zengler, as a professor here at UCSD. And then we have fecal and serum metabolomics and a lot of blood chemistry measurements to allow us to generate these data. This is all linked to the All of Us program, which is doing human genome sequencing. So all of our participants have their human genome sequenced. So this is a hypothesis testing. We use a lot of AI approaches here. So microbiome, metabolomics, accelerometry, surveys, clinical measures, go through a data distillation and the machine learning prediction with things like random forest, gradient boosted regression, and neural network analysis to determine how these features down here, the data, actually allow for the prediction of meal test response curves. Okay, so just to finalize there, what can the microbiome do for us? Well, it can help us develop world-class microbial diagnostics to predict disease and complications before they happen. That's why we need those biomarkers. We need long time series and people to identify the biomarkers which are actually reflective of these diagnostics. We can commercialize intellectual property around microbiome-derived small molecules for the treatment of allergic, metabolic, and neurological disease. So things like indole-3 acetic acid, which I showed you, had an impact upon chronic obstructive pulmonary disorder phenotypes in the lung. They can be potentially used as a treatment once you understand what they do. We can educate on antibiotic stewardship and best clinical practices. Maybe we don't need to pump the body full of antibiotics during surgery, because by doing so, maybe all we're doing is significantly increasing the abundance of organisms that could be dangerous. Maybe before we do that, we should think about giving the people a healthy diet to remove those bacteria. We're redefining that human environmental health nexus to identify the critical inflection points in translational research, where you want to understand how to treat the person and their environment and lifestyle in order to help them achieve optimal health. And we're broadening clinical trials to test the microbiome interventions in high-risk diseases and using new technologies and informatics to support the globalization of diagnostic and therapeutic potential. So microbiome is incredibly valuable for our research objectives. Thank you very much to the people that fund our research and the team of people that do all of the cool work. So this is my team. These are our labs in the background on the beach in La Jolla, and all these people do a fantastic amount of work. Thank you very much.